All right. So the first thing I want to do is thank my co-chair, Nancy Cox. And if you're in the breakout session, group 1, please raise your hand or throw something in the air, whatever. Relax, this isn't going to be a long lecture. We really just have 2 slides. So there'll be plenty of room for discussion. First of all, and not in any particular order, our suggestion number 1 is catalyze the development and hardening of novel technologies and software tools to increase the quality and extent of genome data, importantly at scale. And when we think about the extent, I want to go back and mention a few things. One is structural variation. The other are challenging parts of the genome. Some of them are really holes that we don't really know what's in those sections. The other are just areas that it's hard to map, long repeats. So I could go on and on. I think you get what we mean. The final point would be just the X and the Y chromosome. It's surprising in outside of the Mendelian diseases where inheritance pattern is very informative, how little we know about the X and Y chromosome in terms of genotype-phenotype relationships. The second is champion the inclusion of diversity in sequence-based association studies with a focus on high-impact diseases, and hopefully NHGRI, working with their partner institutes, have co-funding from other ICs in terms of a particular disease phenotype. Again, we spent quite a bit of time talking about what's happening in other domains in terms of sequencing. We have to keep in mind, I think, how Adam started. A lot of the sequencing in the coming decade is really being driven by other programs, by other funding sources, by other ICs, and by other countries, frankly. But nonetheless, there still is a role for NHGRI in particular areas, and I think one area of importance is the role of ethnic diversity, and particularly ethnic diversity in the American population. And by the way, and it's not just for sort of political correctness reasons. There's a lot of sort of novel genome variation because of the history of those populations in terms of discovery. There's things we can discover because we've included various ethnicities that we could not if we just had European chromosomes. And the other is, and sorry to step on other working groups' toes, but as we think about gene by socioeconomic and many other factors and the sort of the social determinants of health, their interactions, it's going to be important to have extreme diversity in the sample set. The other is NHGRI can catalyze and really form a bridge between work that's happening in the Mendelian disease domain and the common disease or polygenic disease domain. Often these areas are going on separately, but there's a lot of complementary information between the two. There's a lot can be learned one from the other. And I think as in some ways, the sort of oligogenic determinants of health have been left to the wayside. That's probably too strong, but are probably understudied because we tend to focus on one extreme or the other. The last bullet on this slide is to create and invest in proximal multi-omics, methylation, long reads, proteomics, et cetera, and do that in cells, in model organisms, and in free-living human beings. And we can use natural and engineered variation and also vary the context. I mean, one of the things that's extremely interesting is to look at the role of genome variation across the life course. That would be one context, and also in response to treatment or environmental perturbation. Maybe this is a little bold, but we decided to label this. We shouldn't talk about the end of the Human Genome Project, but this will be the next phase, really, of the Human Genome Project. And that would have to annotate, and by annotate we mean assign function, do our best to understand mechanism, and really have a broad view of the phenome to all genomic elements. So really take every genomic element and see if we can begin to assign function to that genome element. And by elements, we mean genes, RNA functional units, regulatory units, et cetera. And we would do that by complete sequencing with analysis through understanding the evolutionary context of those genome variations, bring in molecular perturbations, as was discussed this morning, and then have naturally occurred variation in very deeply phenotyped, especially proximal phenotypes, to begin to understand the relationship between genome variation and that phenotypic variation, to try to, again, repeating myself, assign function to every genetic element in the human genome. And then, of course, we would share all of this information broadly and translate it to improve health and lessons about the burden of disease. And one of the things we spent a lot of time in terms of burden of disease is the interoperability between various sources of phenotypes we think would be very important to realize such a program. Okay, so that's really, okay, slides from that group. Any specific comments or clarifications, and then we'll open it up for broader discussion later. Anything I missed from the working group or Nancy?